Good morning, good afternoon, and good evening to all of our webinar participants, and welcome to the third webinar in our series on microplastics and health. My name is Rachel Meidel, and I am a Distinguished Fellow in Energy and Sustainability at Rice University's Baker Institute for Public Policy. I'm a member of the Planning Committee for this webinar series, and also a member of the National Academy of Sciences Roundtable on Plastics. If you're interested in knowing more, the information about this series, including the full Planning Committee roster and the Roundtable on Plastics, can be found on our website. This series is an activity of the Environmental Health Matters Initiative, a forum from the National Academies. This series of webinar intends to provide a solid baseline of information and knowledge to inform future work and decision-making on microplastics. More specifically, the series aims to assist in identifying research gaps and regulatory needs and promote cross-sector dialogue on decision-making regarding microplastics and their potential impacts on human health. So on behalf of the Environmental Health Matters Initiative, and in collaboration with the National Academy's Roundtable on Plastics, I'd like to welcome you to our third webinar in this series that will focus on the potential hazards to human health and the environment from microplastics, and specifically understand the complexity of assessing risks. During the first webinar that we held on February 27th, our speakers and panelists covered methods for sampling and characterizing microplastics as a foundational capability to support exposure assessment and hazard evaluation as we seek to assess potential human health risks. The speaker emphasized the importance of proper sampling, analysis, and QA/QC methods, highlighting the need for collaboration and research and the development of standardized practices across various media. They also concluded that a centralized microplastics database and continued development of ISO and ASTM standards are critical for effective data sharing and informed decision-making. During our second webinar on March 20th, our speakers and panelists provided a standard baseline understanding of microplastics distribution in the environment and the most prevalent sources of human exposure. Specifically, the webinar highlighted the direct link between increasing plastic production and the proliferation of microplastics across interconnected environmental compartments such as air, water, and soil. Key sources of microplastics were discussed, with packaging, particularly food packaging, identified as a major contributor to both chemical leaching and physical fragmentation. Exposure disparities were also emphasized, with vulnerable populations, including children, infants, individuals with health conditions, and communities near plastic production or disposal sites, identified as priorities for further attention. And finally, a precautionary approach was encouraged, advocating for proactive measures to reduce exposure and address potential impacts, even in the absence of established regulatory thresholds, while noting existing data gaps and the need for continued research. Our next webinar will be in May, and we will build on these findings to incorporate in the discussion the consideration for risk communication and the development of exposure mitigation at all levels of decisions, from individuals to federal. Now, that's a lot to cover in the series, but we aim to provide an effective baseline of information to inform future research and solutions through cross-sectoral discussions. The committee would like to emphasize that the content presented in this webinar is not intended to represent the formal perspectives or recommendations of national academies. All views and opinions expressed are solely those of our volunteer guest contributors. This workshop series aims to bring together a variety of stakeholders and experts from different sectors with diverse expertise and experiences. For the workshop, we want to thank the Planning Committee and members from the Roundtable on Plastics for their thoughtful contributions, the National Academy's staff, and today's speakers and panelists, all of whom have worked hard to bring together this exciting event. Now let's turn to today's webinar. We will have 3 presentations to set the stage, followed by a multi-sector panel discussion that will delve a bit deeper into some specific topics and questions. And audience members, please, you are invited to submit questions at any time using the Slido platform. Even if you don't have questions, you can still use Slido and you can upvote any of the questions that you want answered. The link for Slido can be found on the website and in the chat. So with all of that, let's get started. So during this session, our speakers will provide an overview of published studies that have either detected microplastics in various human organs or linked microplastics to health effects. This will be followed by a discussion of established methodologies for risk assessment, examining the complexities, uncertainties, and challenges involved in evaluating these risks and educating the public about them. So it's my pleasure to introduce our speakers in the order that they will speak. Dr. Phil Landrigan, who is a professor, a pediatrician, and directs the program in global health and the common good and the Global Observatory on Pollution and Health at Boston College. Dr. Stephanie Wright, who is a senior lecturer in environmental toxicology in the Environmental Research Group, Imperial College of London. Dr. Todd Gouin, who is an environmental consultant at TG Environmental Research, and Dr. Valerie Hanley, who is a senior toxicologist in the Human and Ecological Risk Office at the California Department of Toxic Substances and Control. As a reminder, all of the biographies for the speakers can be found in the agenda on our website. And speakers will provide their thoughts in brief presentations, and then we will open the floor for questions. And audience members, as a reminder, please submit questions at any time using Slido. And again, if you don't have questions, you can upvote others' questions. So with that, we will start with you, Phil. So if you could unmute yourself and share your screen.
Thank you, Rachel. Here we go.
All right, and then just to keep us all on time, you'll receive a notification in the chat as we reach the end of your short talk. And with that said, I'll turn the webinar over to you.
Thank you very much. Right then. So my topic, hazards of microplastics to human health. So to set the stage, to give a context, a few megatrends. Plastic production is increasing almost exponentially. It's on track to double by 2040 and triple by 2060 if current trends continue. Coupled with that, plastic recovery and recycling rates are very, very low, much lower than I think most people realize. About 8% in this country, under 10% globally. Plastics are very persistent in the environment, including microplastics. They last for years to decades. And because of those three factors, plastic pollution is increasing. There are currently an estimated 6 billion metric tons of plastic waste in the biosphere. That's about 3/4 of all plastic ever made. And because of that increasing accumulation, human exposure is increasing, as Rachel noted in her opening remarks. So here's the trend line, on track to double by 2040, triple by 2060. The biggest part of current production, accounting for the largest accumulation of plastic waste, is single-use plastic, stuff that we use once and throw away, packaging in particular. Human exposure, microplastics are inhaled, they're ingested, and to some extent they get through the skin. This is a slide that was shown in an earlier webinar in the series from Richard Thompson's work, showing that microplastics have now been detected in multiple organs in the human body, basically any organ in which it's been sought. These particles have two main components. I think when we talk about microplastics, we tend principally to think about mass action, a little space occupying object, the polymer. But in addition to the polymer, there are the thousands of chemicals that are put into plastics to give them particular properties, and those molecules are still present in microplastics. And both have the potential to cause harm to human health. The chemicals can leach out of microplastics and can get into organs and tissues, and human body burdens are increasing as pollution increases. Here's a slide from Matthew Campen's work based on brain tissue harvested from dead people starting in the 1990s up to the near present, showing this upward trend in concentrations of microplastic in brain tissue over the past approximately 30 years. The chemicals in plastics are a mixed lot, and they include some very worrisome chemicals, carcinogens like vinyl chloride, 1,3-butadiene, benzene, developmental neurotoxicants that can cause brain injury, especially if exposure occurs during prenatal life, and endocrine disruptors, phthalates, for example, that can interfere with brain development to cause reduced IQ and behavioral problems. and can also interfere with the development of the male reproductive organs, leading to birth defects of the reproductive organs in baby boys and probably decreased fertility in adult life. And it's very important to emphasize that these chemicals are part of the story of the toxicity and the harms to health of the microplastics. These chemicals are in all of us. CDC has a national biomonitoring survey. They find a couple of hundred chemicals, including all of the above ones, in virtually all of us, including newborn infants. And it's very important to remember that while we know a great deal about a small number of these chemicals, we know next to nothing about 80% of them because of the very weak chemical safety laws in this country. Something like 80% of all the chemicals in plastic have never been tested for toxicity, or at least the data are not publicly available. I won't go through this in any detail, but suffice it to say that the chemicals in plastics have been shown to cause disease in both men and in women at every stage of the human lifespan, from premature birth and miscarriage and decreased birth weight, all the way through to increased risks of heart disease, cancer, stroke, and diabetes in elder adults. There's been a lot of attention lately to the cardiovascular hazards of microplastics. There was a paper published a year ago in the New England Journal of Medicine looking at 312 patients in Italy who had carotid endarterectomy, the removal of plaque tissue. This is plaque tissue here on the inside of the carotid artery, and that they took that plaque tissue to the laboratory. They found that 58% of the 312 patients had microplastics in the tissue. Those with microplastics had a 4.5-fold increased risk of myocardial infarction, stroke, or death over the next 3 years. Of course, this is only one study. It certainly does not prove causation, but it's a striking finding. It cannot be ignored. It demands follow-up. We can't simply say that the methods are imperfect and wish it away. This is something that requires further study. Here are some other reported health effects. Increased accumulation in brain. I showed you that slide from Matthew Campion's work a moment ago. Nanoplastics have been found to promote aggregation of a protein called synuclein, which is involved in Parkinson's disease. Microplastics have been linked to gut inflammation. They're linked to preterm birth by way of placental accumulation. They've been found on the testis. These are early data. They're mostly small sample sizes. The methods are not yet standardized, as the first seminar in this series emphasized. But all that said, these data can't be ignored. We can't just say, oh, the methods are imperfect, therefore the data don't count. I think that's the wrong answer. We need to pay attention to these findings and investigate further and either disprove them or refine our understanding of them. So next steps. In research, methodologies are key. Some colleagues of mine at the Minderoo Foundation published a paper just a few weeks ago calling for standardized methodologies, the same call that was made in the first seminar in this series. We need good, high-quality clinical studies, epidemiologic studies. We especially need studies of health impacts in early development, because one of the things we've learned through studies such as the National Academy of Sciences report on pesticides in the diets of infants and children is that even small exposures to toxic chemicals in early development can have severe effects. So studies that simply look at adult impacts are not sufficient. We need to know what happens during embryonic and fetal life with microplastics as particles and microplastic chemicals. In terms of prevention, of course, we have to apply the classic risk assessment, risk management paradigm that was defined many years ago by the national academies. But we need to understand that that classic paradigm has serious limitations, and it really is not very fit for purpose here. The problem is that it's set up to look at one chemical at a time. It is not well suited to looking at multiple exposures that occur simultaneously, which is the nature of microplastic exposure. And it's inherently very, very slow. It can take years, even decades, to do proper risk assessment. And given that plastic production is on track to double in the next 15 years, that's not going to do very much to protect public health. We therefore need to complement risk assessment, risk management, as Rachel said in her opening remarks, with a precautionary approach. And I think specifically, we need to put two things on the table. There needs to be a global cap on production of new plastics. We cannot continue to recklessly produce more and more plastics year by year by year and double production in the next 15 years. It's just not consistent with human health. And we also need to have some rational science-based approaches to regulating the thousands of chemicals in plastics. Chemicals that go into pharmaceuticals get very tightly regulated, and that's because of the thalidomide tragedy back in the 1950s, when between 10,000 and 15,000 babies were born with terrible birth defects following their mother's ingestion of the pharmaceutical chemical thalidomide during early pregnancy. That terrible episode led to the passage of laws in countries around the world, including the United States, that have very strictly regulated pharmaceutical chemicals, require that pharmaceutical chemicals be proven safe and effective before they come to market. I would argue, as a physician who cares for children, that we need a similar regulatory regime for chemicals in general and chemicals that go into plastic. There's no rational basis for strictly regulating chemicals that go into children in the form of a pill, and not equally strictly regulating chemicals that go into children because they come from a toy or from a baby bottle. So with that, I'll sign off and stop sharing and pass the microphone back to you, Rachel.
All right. Thank you, Phil, for your summary remarks about these seminal studies in such a short period of time. And so I will now turn to Stephanie and Todd, who have a shared presentation. And so Stephanie and Todd, if you could unmute yourselves and share your screen and keep an eye on the time, and you'll receive a warning from us that you're approaching the end of your talk. So the floor is yours.
Okay. Thank you very much for the invite to present today. We've got a lot to talk about in quite a short amount of time, so this is going to be a fairly high level just to set the scene for the discussion next. Sorry, get the mouse working. There we go. So as we've just heard from Phil, there have been some really high impact articles recently linking microplastic exposure to health outcomes. This is really reflected in the media, where it feels like every day there's a new alarmist headline. And whilst this is great for raising public awareness of the issue, it's clearly also leading to some public anxiety as well. And so we're going to unpack the sort of data underlying that today. And equally, as Phil mentioned, and the first webinar in this series, one of the issues we're facing at the moment is in relation to the analytical methods. And I think as a scientific community, we're just starting to recognise some of the limitations in these methods, and that would relate to not just the challenges that we face with fighting background contamination in the laboratory, as we've known for quite a few numbers of years, but more recently we've started to learn that small molecules originating from biological material can also interfere with our analysis. And so I think a key message with respect to our needs moving forward and underpinning the rest of this talk is really about harmonisation and sort of really coming to an agreement of what lines of evidence we need for this evidence base moving forward. So we've just heard a bit about what micro and nanoplastic particles are, and I'm sure you have in previous webinars, but just to remind you, broadly we define them as any plastic particle which will include a variety of shapes, including fibres, but importantly they measure below 5 millimeters in their longest dimension. And so as you can see here, this graphic nicely shows the sort of relation of different sizes and different particles. As you can tell from the size range of microplastic and then nanoplastic being anything below a micron in size, this spans a really wide range of sizes, and that can be quite problematic for toxicologists, as you'll hear shortly. And so aside from size being a factor in the definition and the complexity when we start to unpack their hazards, they're also comprised of different plastic materials. They may have different surface chemistries and properties. They may or may not have additives inside. They may comprise different shapes. And this all underpins their hazard, the hazard that they present. And also it leads to a question of what is a representative microplastic when it comes to testing their toxicity in the laboratory. So just to touch on exposure, I know this was covered in a previous seminar, but just as a reminder, there are various routes by which we are exposed to microplastic and sources from which they originate. This can include things like the atmospheric deposition and resuspension from soils, from urban areas, from even the sea interface. Equally, things like sludge spreading are a hypothesized source or mode of transport for microplastics. So there's a wide range by which they may be mobilized and move through our environment and different points at which we might be exposed to them. But really the emphasis is on water, on the diet, and on air. So what we do know, based on hundreds, if not thousands, of measurement studies, observational measurement studies, is that microplastic contaminates the diet, different sources within the diet, and they contaminate air. That's both indoor and outdoor. And so clearly that means that there's an oral and inhalation exposure pathway. Focusing on the oral exposure pathway first, we really know a lot about I'd say our exposure to particles above 10 microns in size. That's because typically studies have used different methods which have a size limitation or a limit of detection of around 10 microns, depending on the technique. And just using this study from Giuseppina Zuri, which was published in 2023, this is a review article which selects publications which used a technique which was able to chemically classify particles in the study. They estimate the daily intake from the oral exposure pathway and the inhalation. And via food, you can see that it's the average adult is exposed to just over 220 microplastic particles per kilogram of body weight per day. Compared to inhalation, it's been estimated in the same study that the average adult will inhale 195 microplastics per kilogram of body weight per day through both their time in outdoor and indoor environments. And then that leads to a total daily intake of a maximum of 417 microplastic particles per kilogram body weight per day, which is just over 30,000 microplastics per day. And these are worst case scenarios based on maximum observational data. But as I said, what's really unknown is the particles in the size distribution below 10 microns in size. And it would be really great if these studies, and especially these reviews which estimate exposure, could even stratify that exposure by size distribution as well. And that's because size really influences how a particle behaves, where it moves into the body, and ultimately its fate. So just starting on the airway, as you can see, particles with an aerodynamic size of around 10 to 30 microns will deposit in the upper airways, so the nose and mouth, and will swallow them. Those that are between 2 to 10 microns in aerodynamic size can travel further down into the large conducting airways, and those that are just below 2 to 2.5 microns aerodynamic size can deposit in these alveolar air sacs, which are down at the end of the terminal bronchioles. So as your airway branches out, right at the end, you get these alveolar air sacs. And this aerodynamic size that I mentioned, it's important because it integrates not just physical size, but shape and material density as well. So it really influences where a particle will go if inhaled. And that's important because then that leads to clearance mechanisms and residence times in the airway. So those that deposit in the large conducting airway will typically be cleared by mucociliary clearance, and again leading to swallowing and oral exposure, whereas those that reach the alveolar air sacs will hang around a bit longer, and they're cleared normally by a macrophage which resides within each air sac. So the clearance in the alveolar can be on the region of months to longer, whereas in the large conducting airways it's usually hours to days. There's a similar story for the gut. So those microplastic particles that we swallow or we intake with food will equally undergo similar mechanisms. So first of all, we also have mucus lining the gut. This has a pore size of around 100 to 200 nanometers. And so you can see already that there'll be a favour for smaller particles being able to permeate that barrier. And typically those with a negative charge more favourably will do so. Once they reach the epithelium, so below the mucous barrier, there are then a series of different cellular pathways by which the particle may be taken up into the body. And you can see here the M cells, which are in the Peyer's patch of the intestines, will uptake particles around a micron in size, but that can range from 20 nanometers to 10 microns, whereas particles above a few microns in size and up to 110 microns may be taken up via the route of persorption. And then there are transcellular and paracellular mechanisms for the smaller particles on the hundreds of nanometer scale. And I should just note here that for the persorption pathway, this is generally a very low rate of uptake. So around 0.002% of particles in this size range will be taken up by this mechanism. And this is generally stochastic, so there's a likelihood to it. So that's, I guess, a bit about the exposure and sort of how these particles will reach cells and interact with these regions in our body. I just want to now touch on some of the hazards. Firstly, it's important to say that we could have direct and indirect effects of microplastic. So a direct effect would be something linked to a physical attribute or physicochemical attribute of the particle directly triggered by that exposure, whereas indirect might be secondary to that exposure. So, for example, if you have localized inflammation, that could progress to systemic inflammation. Then you have localized effects and distal effects. The localized effects will be what occurs at the point of exposure, whereas the distal effects will be what would happen following indirects in secondary tissues. Now I'm going to go through some of the hazards which occur at the cellular level, and just because I'm not sure if everyone online will be familiar with these terms, I just want to give a very basic overview. So we're going to talk about oxidative stress, inflammation, and metabolic activity. So oxidative stress basically is when there are too many free radicals, or what we would call reactive oxygen species, and not enough antioxidants to neutralize it in the body. And these free radicals can be present on the surface of weathered microplastic following the auto-oxidative weathering that takes place upon exposure to sunlight. But equally, they might be generated by the body as part of our innate immune response to microplastic particles. When we talk about inflammation, this is basically the body's response to injury or infection, and this happens because we need to remove damage, causes of damage in the body, we need to clear cellular debris, and we need to repair tissue. So these are the three kind of key things that underpin inflammation. And of course, when we think about microplastics, they're a foreign body, so they may trigger an inflammatory response. And equally, oxidative stress, if triggered by microplastic exposure, can lead to inflammation. Finally, there's metabolic disruption. So this is where in the cell, the normal processes are altered, and that can lead to an impaired function of that cell or even cell death in worst cases. Now, it's been shown that microplastic could disrupt the mitochondria in a cell. So this is the powerhouse of the cell, but equally oxidative stress can also disrupt energy metabolism. So there's some interconnection between these different effects as well. So what have microplastics been observed to do? There are a lot of studies on this, so we've synthesized this for the purpose of this talk. In the airways, it's been found that microplastic exposure in both in vitro cell models, but also in preclinical rodent models, mammalian models, can lead to the formation of ROS and therefore lead to oxidative stress if unresolved. It can reduce cell viability and metabolic activity, increase membrane permeability, and activate inflammatory pathways. In terms of pathology, so tissue level effects and sort of changes to the actual tissue architecture, one of the key outcomes is noted as being fibrosis. So this is tissue scarring and it's irreversible and can compromise function at the tissue or organ level. And important to note is that influential factors related to these outcomes are things like particle shape. So irregular shapes have been found to be more toxic than spherical. Size, whereby generally smaller particles have been found to be more toxic than larger. Surface charge, where positive charges on the surface are also linked to higher toxicity. And then the plastic material itself, but also the model, so the cell line or the organism, seem to have various sensitivities. So moving into the gut, those particles that we swallow can directly interact with the gastrointestinal epithelium once they've permeated the mucous barrier. And there's a similar story whereby we see oxidative stress. Studies have reported observation of inflammation, and other things such as programmed cell death or apoptosis, and the various effects on cell signaling pathways. And so really there's some commonality across these different systems and models with respect to these hazards at the cellular level. So just to finish on hazards before moving into risk with Todd, I just want to bring up this idea of fibrosis again and some old clinical observations from occupational epidemiology. I appreciate new studies on health effects are coming out, are emerging, but there's also an old literature on workers who have or experience very high exposure concentrations. And this is particular to synthetic textiles industries and flocking industries, which cut fibers into very short fibers using a rotary mill. So interstitial lung disease, or we could call it chronic inflammation of the lung, has been observed in the nylon, acrylic, polypropylene, polyethylene terephthalate industries. And specifically, I want to focus on the nylon flock industry. So flock workers' lung is a clinically recognised occupational disease. It's characterised by a chronic inflammation of the lower airways with the inclusion of polyamide particles as well. And importantly, workers with this disease are at a greater risk, workers in this industry are at a greater risk of interstitial lung disease, but also of idiopathic pulmonary fibrosis. So 258 times more likely than a normal population to get this disease outcome. Now, this has mainly been observed in the nylon flock industry, but it's also been observed in a few other types of plastic flock industries as well, although not as frequent. And so this really raises the question about what's driving this. Is it just a sort of overwhelming influx of a persistent material into the airway, or does the plastic material itself really matter, or is it something else, an impurity or additive related to that plastic material? And so I'm just going to end there, and I'm going to hand over to Todd.
Apologies, I had a slight challenge with my controls. So thanks, Steph, and I think that really covers a lot of the aspects in terms of the health effects and also implications towards trying to better understand risk. And I guess it's sort of a big ask to reflect on how we can evaluate the risk for chemicals, or for the microplastic in terms of human health. And when we talk about risk assessment, I guess I do want to sort of take a step back and really think about the fundamentals of what risk assessment are. It's something we've sort of appreciated for quite some time, and I think that's an important observation, for instance, from the 16th century, still plays an important role in how we evaluate risk to chemicals or to any stressor that we might encounter. And Paracelsus, for instance, observed that all substances are poisons. There is none which is not a poison. It's the right dose that differentiates something from potentially providing a benefit to being a poison. So pharmaceuticals, for instance, provide a benefit up to a certain dose. But if you, of course, consume too high a concentration, get too high a dose of that pharmaceutical, it can lead to a negative adverse effect. And so for a lot of things, we want to ensure that we are avoiding situation by which the dose is too high to cause a concern from a toxicological perspective. And so a key component of this within the risk assessment space is to enable us to ensure that we are generating dose response relationships. Can you, next slide, please, Steph? I can't seem to progress this slide yet.
Sorry, Todd, it says you have control of the slide. Oh no, you've dropped it now. Sorry.
Yeah.
Okay, I'm going to share, so you should have control.
Okay.
It says you do have control.
It did before, yes, as well. So the so key component within the risk, of course, is that we have a formalized way of trying to derive a risk, which is sort of relating the exposure to a hazard. And within this context, we want to ensure that we have reliable and relevant data, both in terms of exposure and hazard, and importantly, that we have a dose-response relationship that is based on reliable and relevant information. We have heard in the previous seminars, for instance, in terms of exposure, both in context of what the main sources of exposure might be, but also in the first webinar, sort of the challenges that exist from the analytical perspective and our abilities to provide or to acquire reliable and relevant data that characterize and quantify that exposure. A lot of the data, I think, that are available are, I think, suspect in terms of their reliability and relevance, and there's a need to strengthen that information moving forward, because this is where it really comes into play and to enable us to actually evaluate risk. From the hazard perspective, we would, I think, ideally want to identify what is the primary endpoint of toxicological concern. And I think, as Phil noted, there's a lot of challenges in terms of identifying which is the most relevant or sensitive endpoint that we want to identify and use in this equation to evaluate risk. If we know what that is, I think it is possible to appropriately and sufficiently characterize risk. The other component of this, which I think is really important, is to actually appreciate what are the intrinsic properties of microplastic that are potentially more toxic than others. And this is a component that I think is also important. I think it's unwise to reflect on a thought that all particles, for instance, have an equal toxicity associated with them. It does appear that there are differences in terms of size, differences in terms of polymeric composition, as well as in terms of the shape. And so I think it's important to us to better understand more mechanistically what are those intrinsic properties. As mentioned, we know that there are negative effects. These have been observed in various lab studies. Unfortunately, a lot of these lab-based studies are using non-standard approaches, either in using mammalian in vivo approaches or in vitro-based studies. But some of the observations that we see, nevertheless, are consistent with the results that Steph just showed in terms of occupational epidemiology. We also know that there's an exposure that exists, and overall, for the risk assessment, we need to ensure that data are fit for purpose. I cannot advance slides. I don't know if I did that. So are the data fit for purpose is really important. Toxicity data, the information to date is represented by the use of very high concentrations, which are understood to be inconsistent with typical ambient exposure. And that limited number of studies that we have that do report a dose-response relationship don't typically include an environmentally relevant exposure as part of this component. And I think importantly, the other aspect of this from the hazard perspective is that most of the toxicity studies to date are using a single type of particle of specific size, shape, polymer composition. So for instance, we have a large number of studies that are reporting results for polystyrene spheres of, say, 10 nanometers. And this is not something that we are typically exposed to on a day-to-day basis. And so there's a need for us to ensure that we avoid an apple to oranges comparison in terms of microplastic research. And if in the situation that we have at the moment, there is a lot of apples and oranges comparisons going on, which of course makes it very difficult to do a risk assessment, because we want to effectively ensure that the data we have are consistent between both the exposure and the hazard data that we have. So trying to address this issue, there's some nice research from Bart Coleman's group, and he presented a little bit on this during the last webinar. And what I see in this is sort of an opportunity that exists where we can begin to leverage understanding that exists in other areas of research that have also looked at different types of particles. And one of the important things to reflect upon is the microplastic particles we talk about, that they exist as a continuum in terms of their properties. There is a probability distribution in the environment in terms of the particle sizes, the shapes, surface charge, and ideally it would be useful if we could combine information around these probability distribution functions to different effect mechanisms and use this information as a mechanism to better characterize the risk by considering the whole continuum in which we might be exposed to the different types of particles. We can also leverage, I think, a lot of approaches that have already been developed in the particle and fiber toxicology space, which there are a lot of fundamental best practices in terms of testing materials that exist. And I think these are really important to reflect upon. A key component of this is in terms of ensuring that the particles that are being tested are well characterized in terms of their properties. This was something that we did when we looked at tried to review all of the available data in terms of toxicity studies, and we found that a number of studies did a fairly poor job in terms of characterizing the actual particles they were testing in their systems. More recently, in the EU Horizon 2020 project, as part of the CUSP cluster, a publication from Vogel et al. Also sort of iterates this point, the need to characterize the particles, but also in terms of ensuring that best practices are adopted, such as the adoption of integrated approaches for testing assessment. So there are standard OECD test guidelines, for instance, that exist that should be adopted and used. And ensure that the data that are being generated in this space are reliable, relevant, and that they can be reproduced. Now, Phil mentioned a lot about microplastic and chemicals, and whether or not there's an intrinsic risk is sort of a question I've been reflecting on for quite some time. And I think for me, this does go back to this question of Paracelsus, in that the dose is really important to reflect upon in terms of the risk assessment and whether or not there is a true risk associated with the hazard. This concern has existed ever since the first papers on microplastic were published back in the 1960s and 70s, where there was this a relationship between the chemicals that were in the plastic particles in the environment and a potential for these particles to act as vectors of exposure. In this UNEP report in 2023, I think what's really important here is that they sort of emphasize the need for us to better understand the exposure of chemicals that might be used in plastic throughout the whole life cycle of use. So this would include the production stage, the life cycle use in commerce, as well as the end of life component. I think also important with this is also to appreciate these chemicals that are identified as being used in plastic also have other uses besides just plastic. So we always have an exposure to these chemicals. And the key challenge here is trying to differentiate the relative importance of the microplastic as a vector of exposure to all of the other exposure pathways that might exist. I'm not advancing the slide. There we go. Too many. There's a delay. I have a warning on time, and if only I could advance slides. Okay. This question of the role that microplastic might play as a vector of exposure was also investigated in a WHO report, whereby, again, the one nice thing about chemicals is for chemicals that are of concern, we do have information of the toxicity of these data, and we have established points of departure for a lot of these chemicals, and we can use this information in relation to the exposure that we anticipate for microplastic using a conservative approach, assuming 100% bioavailability, associated with the maximum inclusion levels or maximum concentrations of these chemicals that have been reported in microplastic. The WHO then evaluated what the margin of exposure would be and found that there were no safety concerns associated with this particular exposure pathway. A potential problem with the approach is that it only investigated, in terms of the WHO, it only investigated drinking water as an exposure pathway for microplastic. But of course, these microplastic particles are everywhere in the environment. We ingest them, but other organisms in the environment also ingest microplastic particles. And what I did as part of the European Union project, LabPlas, was to adapt a food web bioaccumulation model that looks at chemical exposure and included microplastic particles as an ingestion for all of the organisms within the food web, and then tried to simulate what the movement of the chemical additives in the plastic would be from this particular pathway relative to different exposure pathways that might exist. Now, a key challenge with this context is actually trying to quantify what the exposure would be for humans, for instance. And again, from a student of Bart Coleman's group, some estimates of exposure have been produced. And one of the things that's really important to bear in mind is that these estimates span more than 20 orders of magnitude, with the median values being around 184 nanograms per person per day for children and 583 nanograms per person per day for adults being reported in this particular study. Now, I used these data to some extent, but also tried to reflect on the fact that you might have the 99th percentile population ingesting very high concentrations of microplastic, which may also have chemicals associated with them. And I used this model then to try to estimate what the margin of exposure would be. And consistent with Paracelsus, then, as the concentration dose of exposure increases, we then have a potential cause for concern. So if we are able to demonstrate that there is an ingestion rate that approaches 10 milligrams per day of microplastic particles that are all 1 micron in size, and that these particles include the maximum inclusion level of these chemicals in the plastic, then there may be cause for concern. But based on the estimates from Bart Coleman's group in terms of the dietary exposure that we have to microplastic particles, it seems that there should not be any cause for concern in terms of this exposure pathway. Nevertheless, it's still important to reflect on coming up with holistic approaches to ensure that we model the fate and exposure of chemicals that might be used in plastic or anywhere in commerce to ensure that there is a safety that can be demonstrated. And I have the last slide, if it advances. Just the outstanding research needs, and this is, I think, consistent with what we keep hearing time and time again. There's a need for standard methods that enable us to generate robust, reliable data. Exposure is probably one of the key components of this that we need to better understand, not only in terms of the analytical component, but also helping to better understand what the sources of contamination are so that we could better direct mitigation strategies to prevent exposure from occurring to especially problematic materials, and to better understand the characterization of these particles that we might be exposed to. This information, of course, from the exposure side could be used to come up with model particles that can be used for hazard assessment. And in terms of trying then to come up with quality assured experiments on the testing side that follow best practices and OECD test guidelines, for instance, in terms of testing the toxicity of environmentally relevant particles. With that, I'm going to stop and move on to, I think, Valerie.
All right. Thank you, Stephanie and Todd, for your discussion on hazard and risks as it pertains to microplastics, as well as highlighting the lack of standardized methods and the different complexities and uncertainties in human and animal studies for measuring microplastics, which can oftentimes lead to inconsistent and unreliable data across studies. So with that, I will now turn to our final speaker, Dr. Valerie Hanley, who will provide a brief overview of actions taken to assess, mitigate, and communicate risks at the state level. Valerie.
Thanks so much, Rachel. So yeah, I'm going to have a big kind of shift in what I'm talking about and look at some regulations that have been put in place by California to try to address microplastics. So again, my name is Valerie Hanley, and I'm a senior toxicologist at the California Department of Toxic Substances Control, which is part of the California EPA. In addition to that, I've served as the co-lead for the Interstate Technology and Regulatory Council, or ITRC's microplastics team for the last 5 years. So I'm going to spend some time today going through California's approach to microplastics, and I'm just hitting the highlights because we've done a lot more than what I can cover in 6 minutes. California has truly been leading the way, I think, globally in terms of moving regulation of microplastics forward. In 2018, the state passed a pair of bills, Senate Bill 1422 and Senate Bill 1263, which have since been incorporated into state law. So 42 is added to the California Safe Drinking Water Act, and it is focused on drinking water and includes included the requirement for a definition of microplastics to be developed, to develop a standard method to apply, applicable to testing for microplastics in drinking water. And it also included a 4-year drinking water testing and reporting requirement and public disclosure of those results. So policy handbook was issued in September of 2022 to help implement that 4-year testing efforts. And the first 2 years of monitoring results is then followed by 6 months of evaluation. And that's going to determine whether or not we need more sophisticated methods moving forward. The other bill that was passed in 2018 was SB 1263. And this resulted in a 2-track approach that is presented in the statewide microplastics strategy, which was issued in February of 2022. And it provides a multi-year roadmap identifying specific actions that the state plans to take. So the first track is focused on solutions. So that's pollution prevention, pathway interventions, outreach, and education. That precautionary approach that we heard about before is super important as we look at microplastics. As the track 2, which is all of this science moving forward, like Todd was talking about, we need to have additional research done on the toxicity of microplastics. And then, of course, we need to improve our sampling and analysis and all of these various other topics. So we need the science and the policy to go hand in hand. So on that pollution prevention, within DTSC, we have our Safer Consumer Products program, and it is approaching regulating microplastics from a consumer products point of view. So in 2023, the Safer Consumer Products program had a workshop that announced our efforts to add microplastics to the candidate chemical list. So a chemical must be on the candidate chemical list for SCP to identify and potentially regulate a priority product containing that chemical. So this proposal has gone through external peer review and is currently in the rulemaking process and is expected to be completed in 2026. So once the microplastics have been officially added to the candidate chemical list, then we can start naming chemical product combinations. And so in advance of that, we are scoping several different product categories that contain or generate microplastics. That's one of our priority products that we're focusing on within SCP at this time. So as soon as we get microplastics added to the CC, we'll be in good shape for that. Now, one of the reasons we have to add microplastics to the CC list is because it doesn't already exist as a chemical of concern by an authoritative body, which is where we generate this candidate chemical list. And that's because there is still so much that is unknown. It is a complex suite of chemicals or microplastics, right? They can be all different sizes and shapes and have different things attached to them. And we don't have them on any particular, list. Like there's not a MCL, for example, a maximum contaminant level in groundwater. So partially in addressing that, just late last year, Senate Bill 1147 was also passed in California. And so this is expanding on the California Safe Drinking Water Act, which was that first one I mentioned, where they defined microplastics and set up this 4-year sampling protocol. And so this bill requires the Office of Environmental Health Hazard Assessment, or OEHA, which is another department within the Cal EPA umbrella, to study the health effects of microplastics in drinking and bottled water, and to evaluate toxicity characteristics and levels of microplastics in water that are not anticipated to cause or contribute to adverse health effects. So this would be then used to set a public health goal, which is the first step we have here in California towards developing maximum contaminant levels, or those MCLs. And if they are not able to set those levels, then they need to identify what data gaps are needed to be able to move forward with setting those public health goals. I do want to talk about the ITRC that I mentioned earlier, the Interstate Technology and Regulatory Council. This is a state-led coalition that brings folks together from all over the United States and internationally. It has state regulators, federal regulators, and then it has industry members and stakeholders that come together in a consensus process to develop documents. So we developed the microplastics technical guidance that is the URL is linked up there on the screen. And then in a second phase, we developed an outreach toolkit. So this is really aiding in that education and public outreach, not just to the public, but also to scientists, regulators, and decision makers on how we can move the needle forward in terms of microplastics research and regulation to ensure that we're getting microplastics the attention that it deserves. And so I really encourage you to check out both of those documents, and I can put the links in the chat for you. But they have a lot of really great information, including data gaps and recommendations for science and how we can get more data that can be comparable to each other to try and set those toxicity thresholds moving forward. And that's it. I just wanted to give a very quick overview of the work that we are doing. And with that, I will hand it back over to Rachel. Thanks so much.
Thank you so much, Valerie, for opening the conversation on what's happening at the state level and also the policy levels. And so I think all of these presentations collectively are going to spark a really engaging and interesting discussion. So we are now going to transition over to our Q&A session, which is going to be moderated by Dr. Margaret Spring. And Margaret is the Chief Conservation and Science Officer at Monterey Bay Aquarium. She's the chair of our planning committee for this webinar series and a member of the National Academy of Sciences Roundtable on Plastics. So Margaret, I'll turn it over to you.
Thanks, Rachel. Wow, this was quite an amazing set of presentations and so much to dig into. So the goal now of the panel discussion, and I think Layla's going to highlight all the panelists so we can have them ready to answer questions, is to highlight the successes and the pitfalls so far in studying microplastics, as well as lessons learned from past research and experiences across the various sectors that can be applied to future work. So the panelists will aim to highlight areas of progress and identify gaps that still exist, which will help inform our next steps in assessing microplastics and human health risks. So thanks again to the audience for submitting your Slido questions. We're tracking them. And so let me start actually with one of the Slido questions. Let's see. So this was covered a little bit, I think, in Stephanie's presentation, but has anyone documented population or community level impacts from microplastic exposure at environmentally relevant levels? And Stephanie, of course, put forward this nylon example, which is super helpful. But of course, people are asking, well, where have we gone with this? So maybe I'll start that with Phil and then turn it over potentially to Todd.
Sure. Thank you. Thank you, Margaret. So the studies that I ran through in my presentation, the cardiac study showing the presence of micro and nanoplastics in the heart and great vessels, the studies looking at microplastics in brain tissue, reproductive organs, gut, and so on. Those are all based on real people. Those are not laboratory studies using high doses. Those are studies based on real people who were exposed to the microplastics in their everyday life. They weren't epidemiologic studies that took a systematic look at levels across the population, the way that the CDC National Biomonitoring Survey measures chemicals in people, but they're clinical studies, and they observed effects at levels at which people are exposed. Now, of course, one can argue that maybe those studies aren't very meaningful because the strategies used for evaluating the particles were not standardized, which is true. But the findings are still very provocative. And for example, how does, if everything is due to laboratory error or some other form of misclassification, how does one expose a 4.5-fold, that's to say a 450% increase in risk of heart disease, stroke, and death in people that had microplastic particles in their great vessels as compared to people who did not have particles? It seems awfully hard to conjure up an explanation for that. Maybe there is one, but I don't know what it is. So I think that we have some signals here, what public health people call sentinel events, early warnings. that have to be taken seriously, and we have to continue to study, but we certainly can't wish them away. And I think that the California approach that Valerie outlined is very important, that the state of California, as they have done so often over the past 50 or 60 years, is moving forward to reduce population exposure to a potentially hazardous substance even before all the I's are dotted and the T's are crossed. It's like in medicine, you know, when a child comes into the emergency room with a abdominal pain, you have to make a decision on the basis of incomplete information whether or not to take out the child's appendix. And if you make the wrong decision, you have very serious complications. And the general posture that we were all taught when we were medical students is, when in doubt, take the appendix out, because the risk of removing a healthy appendix is an awful lot less than the risk of allowing the appendix to fester, burst, and contaminate the entire abdomen. So I think that's where we are today. Information is incomplete, but signals are coming in.
Yeah. And Todd, did you have anything to add to that? I know it was in you're in Stephanie's presentation, and that was super interesting. Anything more?
Maybe if I actually take a contrary position from perhaps Phil's perspective. One is, of course, we're talking about risk assessment here. The other is whether or not there's sort of a moral, ethical component that sort of is a value that we're concerned about. And these are two different sort of questions, I think, to reflect upon. The studies that I think, Phil, you mentioned, the brain study, for instance, has issues from a quality perspective and from an analytical perspective, because it is not clear that they actually are reporting microplastic particles. In that particular study, they report largely polyethylene. And there are data from the analytical side that have shown that there's an interference when they use the GCMS pyrolysis method in that study. There's an interference between polyethylene and fatty acid lipids that are in the brain. The brain, of course, has a high level of fatty acids, has lipid content that's quite high. And it's perhaps not surprising that there is a potential or a very high probability that what they're reporting in that study is not plastic at all, but is something else. If you do the math on that, and I think that was published in this recent communication, that the numbers that they report in that study would be, I believe, equivalent to 5 plastic bottle caps that's in your brain. So this is a plausibility issue that we need to also reflect upon. So it's not to say that, you know, we don't take these things seriously. I think it's important to take it very seriously, but also to reflect on the plausibility of what you're seeing and what the implications are. And again, so risk assessment is one thing, and we're trying to support policy with science-based approaches. But I think some of the messages, perhaps, Phil, that you're also advocating is more a value-laden component. I don't necessarily disagree with, but it's a different approach. And I think it's a different set of arguments that should accompany that sort of perspective. At least that's from my side.
Okay.
To be sure, there's an ethical dimension here. In fact, we held a conference at our institution this past October exploring the ethical dimension of the current situation with plastics. But I'm not talking about ethics. I've studied ethics. I'm talking about data, and I specifically the cardiac study. It's awfully hard to explain that trend. It's also hard to explain the time trend in the brain study. Maybe it's all laboratory artifact. It's possible, of course, but I'd be loath to bet on that.
Okay, well, good. That's a really good conversation. Let's turn to Valerie, actually, because as you sort of sit in the state seat trying to figure out what to do, we did hear that there are adverse effects linked about microplastic exposure and size, shape, and chemical composition, and which includes additives as well as the polymer type influence toxicity. So are there any priorities among the microplastic types relevant to toxicity that are being looked at for research or policy attention, sort of like the top list that is sort of coming to the top of your sort of attention in the state perspective?
Yeah, I mean, I think that microfibers are something that are really coming up. Fibers, just because of their shape and the length of them, and it's kind of one of the things that we're seeing the most in water in particular as well. So I think that there is a lot of focus on those fibers. But from, I think it is such an interesting conversation because, there's the folks that are trying to look at all of the studies that have been done, and like what Todd is saying, and really get an actual toxicology type approach to it. And there's still just so much information out there. I do think that as was shown, I think in one of Stephanie's slides, you know, the smaller particles are an issue, but they're also harder to measure and analyze. And so there's so many different things. So I think that that's why, in terms of regulating the products, we're kind of taking a wide range of look and trying to just reduce that. But from a toxicology standpoint, there's just so much information out there, and it's so varied. And trying to get that data to align and be presented in a way that's comparable and that we know there isn't interference is just incredibly challenging. And I know that's not a satisfying answer, but it's the truth of what we're dealing with this complex issue.
Okay, well, thank you. So many things to ask. Can we just dig back into maybe Stephanie? You touched on this, but how exactly do microplastics interact with cells and tissues? Is there anything more to add to this? For example, how readily do microplastics cross biological barriers, such as lung tissue or the placenta? Also, the question of we talked about last time, which was packaging and dermal exposure, and how they accumulate in different organs. You know, what factors control this process? I might ask Stephanie and then maybe Phil to follow up on that.
Sure. So, I mean, in the gut, it should be quite hard theoretically, because we've evolved, you know, alongside particles in our environment, and our body should be keeping them out. Of course, there are always exceptions. In the gastrointestinal tract, the main uptake occurs in the small intestine, in the duodenum, and there are some specialized regions, particularly called the Peyer's patches, which contain cells that are highly specialized for engulfing microparticles from the overlying lumen. There are some size restrictions as to what they do actively engulf, and then for the rest of the cells in the surrounding tissue, the enterocytes, it's generally particles below a couple of 100 nanometers that are favorably taken up, and then there are even smaller particles will passively cross those cells. But I mean, based on the data or size data so far for microplastics in the diet, the more relevant pathway is that of persorption. Persorption, however, you know, it has a very low rate. So for every you know, like however much you ingest within that size range, just 0.002% of those particles are predicted to cross via that mechanism. So it's like a low likelihood, I guess, is what I'm trying to say. The lung is slightly different in that we have a mucociliary escalator to clear particles that deposit in our conducting airways, and those particles that reach the alveolar are likely to be engulfed by a macrophage and cleared either via, I guess, clearance to the lymphs and drainage, or depending on where it is, up the mucociliary escalator.
Okay, great. Actually, Phil, I might ask you a different question because we're running short on time, and I wanted to get at this. You touched on it. We talked about these approaches of risk assessment, mostly focusing on exposure, quantity, and you touched on this, but as we push into younger life history stages, timing also seems to be at least as important as quantity. Can you give us a sort of a short and concise answer around that question?
Yeah, and this is information that comes initially from studies of humans and has subsequently been backed up by toxicologic investigations in the laboratory. But we've come to realize that even very brief low dose exposures in the early stages of human development, which basically means during pregnancy, and especially the first trimester of pregnancy, can have long-lasting effects, effects that would appear to defy the classic logic that traces back to Paracelsus that the dose makes the poison. I mean, generally speaking, of course, the dose does indeed make the poison, but what we've learned from episodes like the thalidomide episode or prenatal lead exposure or prenatal exposure to organophosphate pesticides, is that even low-dose exposures, brief exposures in early pregnancy can damage developing organs and cause lasting damage. And the classic case has always been the thalidomide tragedy, but it's been replicated with multiple chemicals since that time. So I've been arguing for years that if we really want to protect population health against anything, whether it's microplastics or lead, you need to set exposure standards at low enough levels to protect the most vulnerable. And if you protect the most vulnerable, then of course you protect everybody.
Okay, well, great. Thank you. Some of these questions are, we could spend hours on. I'm trying to figure out how to ask one, but let's try Todd. You touched on this, but let's dig into what we know about concentrations at which biological effects are observed in the lab and how those findings translate to real-world environmental exposures. Is there something more you can say about that than you were able to cover in your presentation?
Well, I think there's two components. It's not there's concentration. So we have in terms of exposure, I think relatively not the best understanding of what that exposure is. So I think as I showed with Bart Kuhlman's group, we're looking at more than 20 orders of magnitude in terms of estimates of what the exposure to microplastic particles might be for humans via inhalation, dietary exposure pathways. The concentrations people are using in the lab tend to be very, very high. And so on the order of, off the top of my head, I want to say something like 20,000 particles per milliliter, for instance, in drinking water studies to rats. And this is, of course, again, the key component of this is that people are using polystyrene spheres, for instance, or a single tip of particle, which is not representative of the exposure that we have in the environment. So the challenge here is you say microplastic. Microplastic in one study is not the same microplastic in a different study. So we're not all talking the same language. And I think we have a problem with definition in this context. And so that needs to be clarified, I think, to a significant extent. Groups are trying to generate their own particles in the lab, microplastic particles as model particles to be representative of environmental exposure. And I can say that there is a real challenge actually in generating particles, particularly the smaller size particles, less than 1 micron in size. And those are the particles, I think, as Steph pointed out, tend to be the ones that have the potential to translocate and potentially are going to have a higher potency associated with them than the larger particles. So there's a combination of factors, not just the concentration perhaps, but the fact that what we're doing in the lab has no resemblance to what is occurring in the real world. And it's just not possible to do a risk assessment because it's an apples and oranges exercise.
Okay, thank you. So we've got about 5 minutes left before the final question. So I'm going to turn this question to everybody who wants to answer. What critical data gaps exist in linking microplastic exposure to human health outcomes? It's a hard one.
I think the biggest problem is we just know so very little about these particles. We're in a very difficult situation, what the great Michael DePledge in the UK calls a wicked problem of having so many unknowns in the face of escalating exposure that it's hard to know where to begin on the research. Of course, we will begin because that's what the scientific community does. But I think even as we move forward with the research, we must also be moving forward in parallel to cap plastic production and to limit the number of toxic chemicals in plastic, because we need to go upstream here. We can't just be dealing reactively with very small problems of dose and concentration. This is a public health crisis that has been beneath the radar up until now, but it's coming into prominence, and we need to address it the way the state of California, for example, is doing.
Are there others?
I was going to say, when you say health outcomes, health effects, I always think of, population level patterns and epidemiology. And for epidemiology, we need exposure estimates. And for exposure estimates, we need sound understanding of concentration and you know, then the models to model that exposure. And hearing from Phil, then I guess we need that for vulnerable populations. That would make the most sense for me. Some more evidence on exposure and exposure estimates that we're then able to link to health outcomes in those populations.
Yeah, and I would add, I think that's the great thing that California's been doing in terms of the Safe Drinking Water Act, right? Because there's been this ongoing effort to measure how much microplastics we have in our drinking water. And then now in this next phase to see if we can develop screening levels that are health protective. But at least that gets to the exposure piece of it once we have all of this years of collection data that we're going to have, and that we can go forward. There's been a lot of this done in the aquatic toxicity. So in terms of like, in the oceans, and a lot of sampling, especially in the San Francisco Bay Area. And I see your Monterey Bay Aquarium on your background, Margaret. And certainly there's been a lot of microplastics being analyzed in the Monterey Bay as well. And so the more we know what's present and what populations, not just human populations, but even, you know, when we know what's happening at certain levels, I think that's really going to help us move forward in terms of the exposure. So I think it's really both of those things of let's make sure we know what the exposures are, but let's also do what we can to prevent those exposures from increasing as we move forward.
Great, thanks. I don't know if you want to add anything, Todd, before we go to the last question.
I think sort of reflecting on Phil's comment, you know, plastic pollution is perhaps the problem that we're sort of addressing in a larger context. And I appreciate the suggestion that the idea of capping production is perhaps suggested as a solution. I don't know if it's the best solution, because it would be benefit from science-based information or evidence to help us understand what are the key sources of plastic pollution into the environment, so that we could better manage that waste and introduce strategies to mitigate the exposure from occurring in the first place. So sort of stepping back and actually looking perhaps at the problem on the plastic pollution side in terms of where this is being generated. And I think that's actually a solvable problem, and something I'm more optimistic about. I agree that if we're going to focus on risk as sort of an answer to helping to inform things moving forward, that it will take a lot of time for us to get the right data. But if we just step back and try to focus on the plastic pollution issue and appreciate the solid waste component of it, I think that would really provide a great advance in terms of addressing the problem.
Yeah, that sounds like that's what California did, prevention. They're reducing plastic production is one of the primary areas for that. So, but also it's in cleanup and prevention for sure. So thanks for that. So our last question is sort of to all of you again, and we've got a little bit of time, but maybe 5 minutes. When it comes to microplastics and improving research and the ways we assess or address the risks to human health, in 1 minute or less, can you tell us what you see as the most critical needs and the most promising path forward? I think you partially answered that, but if there's anything more to say about the exposure, more than exposure data, that would be the time to ask to say it. Thanks.
So the Achilles heel of environmental epidemiology is always exposure assessment. You can't assess the relationship between an exposure and an outcome, or a dose and an outcome, if you don't know the level of exposure. So the first need from a scientific point of view, as Todd has said, as Valerie has said, as Stephanie has said, is to come up with standardized metrics for assessing human exposure to microplastics in drinking water, in food, in the environment generally, and probably also for assessing levels of microplastics in human tissues and human body fluids, so that we can measure internal exposures. And that needs to be coupled with standardized measures for the various chemicals that are delivered by plastics, maybe not exclusively from plastics, but primarily from plastics like phthalates. We know that they're bad actors, and we have to have standard measures for the chemicals, even as we're looking at standard measures for the particles themselves.
Maybe Stephanie?
I completely agree. I think it comes back down to the exposure and the exposure assessment and advancement there, because I think there are, we have quite a lot of hazard data. It's just difficult to make sense of it, as Todd said, with this kind of apples and oranges extrapolation. So yeah, completely agree. Better improved measurements and exposure.
Valerie? Mhm.
Yeah, I wanted to give a quick plug, shout out maybe to Tomex, which is the Toxicity of Microplastics Explorer. I know you guys had Leah Thornton-Hampton on previously, and she's led that effort here in California. But that really works on trying to get the toxicity data into one platform so that you can compare across different studies and try to have some correlation between them. So I think that work both on the aquatic side and the human health side is really important and really critical for people to look at. It's hard to keep up with because, people have to go and mine all the data, put into the system, and that takes a long time. And, we know that the studies are just increasing exponentially as the microplastics are, increasing exponentially. And so that is all really challenging. And I guess the other thing I just want to say is I think the biggest hurdle is that microplastic is not a microplastic is not a microplastic. And I think that is the theme that we've heard over and over again. There's size, there's shape, there's what's adsorbed to that microplastic. And so unlike a single chemical where we can do all of these tox studies and say, here's the exposure and here is the hazard that comes to that, here we have this huge variety, this huge group of things that have all kinds of different I'm spacing on the word I want to say, but they're all very different, right? Physical characteristics and everything. And so that makes it really challenging in the risk assessment point of view to try to take this huge group of chemicals, right, as we're calling microplastics, and have one criteria that comes out from that is health protective. So I think that is one of the huge challenges, and again, why that precautionary approach of trying to reduce while the science catches up is critical.
Okay, thanks. Thank you. Very useful. Todd?
Completely echo perhaps everything Valerie said, Steph said. Phil, exposure really important, and these other aspects in terms of definition and trying to focus on what perhaps is most important and what really matters from the exposure perspective. I would say from a funding agency, anybody wanting to fund, there seems to be a trend to want to fund the risk assessment side already when we don't actually have standard analytical methods. So again, when people are analyzing complex biological matrices, even complex food and beverage matrices, we do not have good analytical methods. And there's a high probability that what we're getting is not reliable and relevant information. And there's a danger in that information because it does create, I think, an alarmist potential messages and misdirects perhaps where resources should be invested in. And so if I was going to recommend anyone who had money to spend, please spend the thing that's least sexy, which is do some analytical work, fundamental analytical work, so that we can get a better understanding of exposure.
Well, great. I think we could have a whole day of conversation around these issues, and I'm sure your days are filled with that. So thank you for sharing your time with us. I think what we heard here, which was super important, is that there's a lot of convergence around the need for more science and to take this seriously. That precautionary approaches could be employed, but there are some real barriers to our knowledge. And so I think that there's a lot more to be done here, clearly, and we really appreciate your perspectives from the medical to the practical at the state level. And I'll turn this back over to Rachel for the close. Thank you.
All right, thank you, Margaret, and thank you to the speakers and the panelists for your thoughts. It's been a really great, productive, and informative discussion. And thank you also to our attendees. We had a lot of questions, and we could not answer all of them, certainly, but we collected all of your inquiries, and we will consider them as we prepare for our next webinar in this series. So I'll try to summarize what we've covered, and we covered a lot. There is a need for accurate and standardized analytical approaches to ensure accurate identification and quantification of exposure in relation to hazards. Size, shape, other factors are important in terms of impacting exposure and the types of hazards that may be observed. Hazards may be direct effects or indirect effects. There's a need to translate the effects observed in higher dose laboratory studies to the exposures to human populations. Occupational epidemiology studies represent a source of information for understanding potential hazards in relationship to exposure levels. There are an increasing number of studies identifying the presence of microplastics in human organs and potential harms to human health. And this has raised concerns for the public and scientific concern, and these concerns are not abating. There are chemical additives in microplastics, and these might contribute minimally to overall exposure risks, even under the worst case ingestion scenarios. But we need to know more about both the levels of the exposure and the mechanisms of impact across these pathways. Exposure does not necessarily equate to adverse health effects. That was something that was underscored. Advancing our understanding of microplastic health impact, it requires standardized methods for both exposure assessment and toxicity testing. And there's a need for interventions and improved understandings across the entire life cycle of plastics. There's also a need for rigorous, high-quality clinical and epidemiological studies and data that is fit for purpose. And there must be a bridge for science and policy to work hand in hand. And so all of this is going to help us in our next webinar series when we talk about risk communication. So as we wrap this up, the next webinar in the series is going to examine the critical role of risk communication in considering exposure mitigations to protect human health. And we will build on the discussion from today and from the past two webinars in the series. And this webinar is currently planned for May, and you will be able to submit your questions as soon as the registration opens. And so if you want to stay informed on these future webinars and EHMI activities, please subscribe to the Environmental Health Matters newsletter on our website. And finally, your voice matters. So please take a few minutes to complete this post-meeting questionnaire, and this will help inform this series and our future activities. So thank you again, Valerie, Phil, Stephanie, and Todd, for a very stimulating discussion. And we hope to see you all in May. This webinar is now adjourned. Thank you.